afternoon, everyone. So my name is Saghai. I'm a biologist, and I'm going to present you the work I've done during the first year of my PhD, where I've been interested in the characterization of microbial taxa that could potentially contribute to carbonate precipitation in modern microbialites. Microbialites are defined as self-sustained organosedimentary structures. Self-sustained because they harbor a high diversity of microbes with a huge variety of metabolisms which result in the recycling of various elements. And they are organosedimentary structures because there is a trapping of sediments in the matrix produced by the microorganisms. And that in addition of this trapping of sediments, there is also lithification that is occurring through mineral precipitation, and this mineral precipitation is influenced by the microbes that inhabit microbialites. And you know, of course, the stromatolites, which are actually one type of microbialite. So microbialite is the general term for these structures. Microbialites are interesting if you want to study mineral precipitation, but they are also interesting in an evolutionary perspective because they are very abundant in the fossil record, and they are the oldest, well, today they are the oldest trace of life that we have. It's dated at 3.4 billion years old, so it's very ancient. And you can see on this graph that they have been very abundant throughout the history of life, with a peak at the Proterozoic, and then a decline to the present day. And so today we've talked about a lot about Metazoa, the Cambrian explosion. And you can see it's here, but you have all this part of the history of life that we know very little about, and microbialites are a very relevant source of information. So, as I said, today they are restricted to a few environments where you have specific conditions, environmental conditions, that are favorable to the precipitation of minerals. So these conditions are high alkalinity and desaturation in specific elements such as calcium or magnesium. Microbialites are found in both marine and freshwater environments. This is an example from the Bahamas, and this is another example from Lake Alchichica, and this is actually the lake I've used as a model in my study. And even though you find microbialites in both environments, the processes leading to their formation are different. Very simply, the main differences is that in marine environments, the accretion of small particles is the driving force because of the waves and the tidal forces. Whereas in lakes, in freshwater systems, the in situ precipitation of minerals influenced by some microbial activities are the dominant processes. And that's why, as a biologist, I'm more interested in lakes than in marine environments. So to have mineral precipitation, you need specific environmental conditions, and the biological activity can influence these environmental parameters. Usually, the microbes that you find in microbialites are classified into 6 categories, depending on their metabolic activity. 3 of them are known to promote precipitation, and 3 of them are known to have the opposite effect. I'm only going to talk about these 3 metabolisms. Actually, it's 2, because it's photosynthesis and sulfatoreduction. So you have the oxygenic photosynthesis performed by cyanobacteria and also eukaryotes. And you have the anoxygenic photosynthesis that is performed by various bacterial taxa, such as alpha and gamma proteobacteria or chloroflexi. So during the day, you have light, so you have photosynthesis going on, and oxygen production and local increase of the pH. And at night, when there is no more light, you don't have photosynthesis, so you can have the anaerobic metabolisms that can act, that can be performed, and this is where sulfate reducers come into play. So all day long you have processes promoting precipitation that go on in these systems. I've talked only about bacteria because they have been quite actively studied in currently living microbialites. Today we know that archaea are very scarce, so they do not play an important role in microbialite formation. So this is today's situation. Maybe in the past it was different, but we don't know. The microbial eukaryotes are present. We strongly suspect that they are important, but they are not very well characterized. One of the reasons is that historically bacteria have been more appealing in this field because the earliest microbialites, those which were formed before the emergence of eukaryotes, so before 2 billion years ago, were exclusively formed through the action of bacteria. So from 3.5 to 2 billion years ago, there were no eukaryotes. So that's why, in an evolutionary perspective, people study more bacteria, even though today they are both eukaryotes and bacteria. So it's a bit strange. Back to the lake. So Lake Alchichica is located in Mexico. It's an alkaline lake with two dominant mineral phases, hydromagnesite, which is a hydrated magnesium carbonate, and aragonite, that we have heard a lot about. But actually, hydromagnesite is much more abundant, mainly because of the ratio between magnesium and calcium. That is, well, there is much more magnesium than calcium. There have been some preliminary studies over the last few years on this lake. These studies were done on a small scale, but the microbial diversity was characterized using PCR amplification followed by cloning and Sanger sequencing. It was found that bacteria were very diverse, that archaea were very scarce, and that eukaryotes were significantly abundant and quite interesting in a phylogenetic point of view, as we shall see a bit later. What was also shown in these studies is the existence of calcification mechanisms that were previously unknown. One of them that you have heard about probably is the intracellular calcification occurring in one cyanobacteria called Glomargarita lithophora. This cyanobacteria produces vesicles of calcium carbonate in its cytoplasm. But what is more interesting to me today is the extracellular calcification mechanisms occurring in another cyanobacteria that belong to the procapsals. Here on the picture, you can see a fossilization gradient with here the living cells and the dead cells here. And what you see in gray is aragonite. And what they found is that a colocalization between aragonite and procapsals, so a specific association between the two. It was further evidenced by this picture. It's an autofluorescence picture. You can see here the living cells, and maybe you can distinguish here some remnants of fluorescence, which correspond to dead procapsals that are encrusted into the aragonite. And what they found is that procapsals are specifically associated to aragonite, and that they actually are directly involved in the precipitation of this mineral. Because one of the reasons being that they have a unique F layer, so it's around their cell, that binds specifically calcium over magnesium. And so even though you have less calcium than magnesium, you have a nucleation center for aragonite. So this was very interesting because it showed that cyanobacteria or microbes in general do not only influence the environmental conditions, but can directly drive the precipitation of a specific mineral. So as I said, these studies were performed at a small scale. They were very interesting, but they were preliminary studies. So we wanted to do a macro scale study for several reasons. The first reason is that we could have a deeper characterization of the microbial communities. using a high-throughput metagenomic analysis. We could also, and this is the very important point of my talk, quantify the relative abundance of taxa potentially involved in carbonate precipitation. And finally, we could test at the microbial scale whether procapsules are actually associated to aragonite. To do so, we sampled the lake in 2012 on 2 sites. And the first site was sampled at 4 different depths, so along a depth gradient from 1 meter to 15 meters. And this site was chosen because it was close to the site that was used in the previous study, along a depth gradient as well, so we could compare both results. And we also sampled the northern shore only on the surface this time to check for medium scale heterogeneity, because these environments are very heterogeneous. So from one microbial to another, you can have very different aspects, mineralogy, and some microbial communities. So the idea was to have a better view of the diversity in the lake. We used a metagenomic approach. Metagenomic means that you work with the environmental DNA without any amplification step before sequencing. So we had our 4 samples from the depth gradient, and also the northern sample that we sequence in triplicate, so 3 times. You generate a lot of sequences when you do metagenomics, so from around 100 million sequences. And from these sequences, you can mine for genes corresponding to a phylogenetic marker. So the one that is widely used is the gene corresponding to coding for the RNA of the small subunit of the ribosome. So it's a universal marker. And so you can have access to the members of the 3 domains of life. And this is very interesting because you have this information in the same from the same sequencing step. So you can do semi-quantitative comparison between the abundance of bacteria and the abundance of eukaryotes. And this was not possible with the classical methods, because when you use PCR amplification, you use primers that are specific to one domain, for example, bacteria and eukaryotes, and then you cannot compare the abundance of the 2. So this is very, very interesting. And if you have to remember one thing for this slide, it should be this semi-quantitative information. So once you have predicted the presence of the genes, the fragments corresponding to rDNA genes, you can find to which group they are affiliated by BLAST using reference databases. And you can see you end up with a lot of sequences compared to other methods. From this, you can perform microbial diversity surveys and so have access to this semi-quantitative information that is so important to me. And in the end, you can try to identify and quantify taxa that are potentially involved in carbonate precipitation, for example, using multivariate statistics. So this was for the biological aspects. We also had information on the mineral composition of our samples, thanks to a collaboration with Karim Benzahra and especially Nina's work. Merci, Nina. That we can use later, as you shall see. So these are the results. So you will see several graphs like this one. So the first 3 rows correspond to the northern sample, and then you have the eastern sample with the depth gradient. This represents the relative abundance of sequences affiliated to the 3 domains of life. As you can see, bacteria are very abundant. They represent 90% of the sequences. You can barely see archaea on the right because they are very scarce. So they're only less than 1% in each sample. But interestingly, eukaryotes are significantly abundant. They represent from 5 to 10% of the total number of sequences. And again, this is one of the first, or maybe the first time that in the same study we could compare the relative abundance of eukaryotes and bacteria. If we look closer to eukaryotes, you can see that the green color dominates. The green color correspond to photosynthetic groups. They represent up to 90% of the microbial eukaryotes. We've removed the animals. And also you can see several colors corresponding to various groups, and actually all major eukaryotic groups are represented. So they are very diverse, even though they are dominated by photosynthetic organisms. And if you remember that photosynthetic organisms can influence the precipitation of minerals, you can imagine that eukaryotes could have an impact. The northern sample is dominated by diatoms, whereas the eastern sample is dominated by green algae. So you can see the heterogeneity between the samples and the sites in the lake. This is the same figure, but for prokaryotic diversity. 3 groups dominate: the gamma protobacteria, alpha protobacteria, and cyanobacteria. So all cyanobacteria are photosynthetic, and there are several members of the gamma protobacteria and alpha protobacteria that are also photosynthetic. So in bacteria as well, the photosynthetic organisms could be very abundant, or are abundant. And you can see again that there is a very high diversity with all major bacterial phyla that are represented. Now, if we zoom in cyanobacteria, you can see that the picture again is different from one sample to another. On the northern one, the oscillatorials dominate. These are filamentous cyanobacteria, whereas in the eastern sample, diversity is dominated by Procapsa. So these procapsals, as you can see, they show a pattern. They increase in relative abundance with depth. This was already shown in a previous study. So we confirm the result from this study. And what you can notice here also is that the yellow color corresponding to chlorococcals, well, these organisms seem also to show a similar pattern of increase with depth. So going from this data on the cyanobacterial diversity, we wanted to know if we could correlate this pattern of diversity with the elemental composition of the microbialites. And to do so, we conducted canonical correspondence analysis using 2 matrices, one being the cyanobacterial orders frequencies that I've just shown you, and the other being the elemental composition of the microbialites. You can see that this axis represents 97% of the variation, so it's very informative. And if you look on this side of the graph, you can see that procapsals correlate positively with calcium, which confirm that there is an association between procapsals and calcium, but also, and that was not really expected, chlorococcals also behave similarly. So this was a surprise. And if you look at the same data, but not with the elemental composition, but with the mineral composition, so it's basically the same graph, but differently, the same information, but shown in a different way, you can see that chlorococcal and procapsals correlate with aragonite. The other orders of cyanobacteria are not very interesting regarding carbonate precipitation. So this led us to the hypothesis that chlorococcals could be, as procapsals, be involved in the precipitation of aragonite. Last but not least, the potential microbial contribution to carbonate precipitation is shown on this figure. So these are pie charts representing the proportion of these taxa regarding the total number of sequences of reads. As you can see, they represent from 20 to 40% of the total number of reads, which is huge. You can also notice that the blue color dominates, it corresponds to cyanobacteria. So it was known that cyanobacteria were the main drivers of carbonate precipitation. But here, what we show is that anoxygenic phototrophs, sulfate reducers, and photosynthetic eukaryotes are also potentially important and significant contributors. And microbial eukaryotes are particularly interesting because while they represent only 10% of the total number of reads, they represent in some cases 25% of the taxa potentially contributing to carbonate precipitation. So they could be. So this challenges the classical view, proposing that cyanobacteria are really the main drivers of carbonate precipitation. A few conclusions. First is that the more quantitative information provided by the metagenomic approach allow a better characterization of complex microbial communities, because these communities are very diverse, as you have seen. What I've just said is that taxa other than cyanobacteria might also largely contribute to carbonate precipitation. This is quite new, the confirmation of a suspicion we had. And finally, that procapsules are actually involved in the precipitation of argonite. We've confirmed small-scale studies with this metagenomic study, and that chlorocalcales could as well be involved similarly to procapsules. Thank you for your attention.